Scintilla Biomark Private Limited, Bangalore-based molecular diagnostic company, offers R&D in molecular profiling of chronic human diseases including cancer, cardiovascular, metabolic, inflammation, infectious diseases, and high-throughput metabolics, biomarker validation, and regulated bioanalysis. Scintilla Biomark aims to improve human quality of life by offering excellent clinical and molecular diagnostic services to patients for timely and potentially life-saving therapeutic interventions. Scintilla seeks to develop molecular diagnostic kits for accurate diagnostic and prognostic molecular tests to the healthcare providers in a timely and accurate manner in the following areas of diseases: cancer diagnosis, prognosis, metabolic disease diagnosis, prognosis, infectious diseases, cardiovascular diseases diagnosis and prognosis, anticoagulant therapy monitoring, tissue typing for transplantation and monitoring, universal genetic testing, fertility tests, prospective parents testing, consanguineous couples, CNS disorders, bone marrow disorders, cystic fibrosis, osteoarthritis, thalassemia, clinical research, biomarker identification and validation. We offer internship and training for medical, science, and engineering students as well. Dr. Nagendra is the founder and MD of Scintilla Biomark Private Limited, and he holds a position as Director of BGS Biomedical Research Institute and Scintilla Academy. In his previous positions, he was Director of Pediatric Oncology and Molecular Pharmacology at Memorial Health University, Savannah, Georgia, United States, and Clinical Development Program at Dr. Reddy's Laboratory, Hyderabad, India. He has undergraduate science degree, Masters in Biochemistry from Mysore University, and received PhD from National Institute of Mental Health and Neurosciences. After moving to the United States in 1993, he earned a Master of Business Administration degree from Mercer University, Atlanta, Georgia, and performed his postdoctoral research in pharmacology and toxicology and medicinal chemistry at the University of Kansas, United States. Dr. Nagendra then worked at Cedars-Sinai Medical Center in Los Angeles, USA, where he was a group leader and principal investigator in brain cancer and stroke research. He later moved to Vanderbilt University as director of the Neuro-Oncology Research Laboratory. He has filed 12 patents from United States, European Union, and Japan. He has over 100 peer-reviewed publications, including several book chapters, and presented in over 100 national and international scientific meetings. He had received research funding from the National Institute of Health, National Cancer Institute, American Cancer Society, Georgia Cancer Coalition, and many industry sponsors.
Our institution also has a library, a conference room, and a training room where we give training for interns who come to our institute.
The Olympus BX43 trinocular research microscope with ASI spectral imaging software is used for karyotyping. We use this instrument to identify numerical and few structural abnormalities by peripheral blood lymphocyte culture from patient blood samples. Real-time PCR is a technique used to monitor the progress of a PCR reaction in real time. At the same time, a relatively small amount of PCR product can be quantified. This technique is based on the detection of the fluorescence produced by a reporter molecule, which increases as the reaction proceeds. In our laboratory, we use it for research and training.
FACS TM loader that helps in rapid analysis of multiple cellular processes. It can analyze a variety of cellular processes including apoptosis, cell signaling, DNA damage, cell cycle proliferation, and viability. We use it in our cancer-related studies, microbial and viral analysis, and also for research training.
7500 Fast DX real-time PCR with STS software from Applied Biosystems is a real-time nucleic acid gene amplification and 5-color fluorescence detection system available for in vitro diagnostics, molecular oncology, and microbiology research. This instrument delivers the performance required for high-quality results in a 96-well format. It also minimizes the effort required to perform sample setup while also allowing the use of tube strips. The 7500 Fast DX instrument sequence detection software module offers the flexibility to develop assays and also run pre-optimized protocols for users operating in a secure environment.
Enzyme-linked immunosorbent assay instrument is from Thermo Scientific called the Multiscan Go. It is capable of high-throughput screening and detection of antigen antibody in clinical samples in a 96-well format. For example, we show you CA125 antigen, a marker of ovarian cancer in patient samples.
The Olympus Phase Contrast Culture Microscope CKX53 helps in improved imaging and usability. It also facilitates visualization of cell morphology. With improved imaging quality and handling, the microscope helps in a variety of cell culture needs including live cell observation, cell sampling and handling, image capture and fluorescence observation. Thermal cycler is used to amplify DNA or RNA by PCR method. It is useful in both laboratory and clinical settings, including DNA fingerprinting, detection of bacteria or viruses, diagnosis of genetic disorder, and preparing samples for DNA testing. We are using this instrument to identify genetically inherited iron deficiency anemia in patients. The Hydrosis 2 scan is an IVD-certified semi-automated electrophoresis system from Sebia. There are 48 inbuilt migration modules. We assay hemoglobin and detect major hemoglobin variants like S or D and C or T. We also analyze protein in patient serum and urine samples. Agarose gel electrophoresis separates DNA or RNA fragments according to their size. It uses an electrical field to move the negatively charged DNA through an agarose gel matrix towards positive electrode. Shorter DNA fragments migrate through the gel more quickly than the longer ones. Once we complete the PCR process, electrophoresis is performed to visualize the DNA or RNA fragments. After the gel run is completed, the gel is viewed under UV transilluminator. We perform wide range of cell culture studies using primary and established cell lines. The Biosafety Level 2 laboratory is used for culturing work. CO2 incubator is used to grow cells. We have a liquid nitrogen tank to store cells and clinical samples. The lab has -20 and -80 deep freezers to store the reagents, media and cryophilized cells.